Okay, so we will discuss each one by one. First, we will discuss about single diffusion in one dimension, otherwise called as Auden procedure. So here, single diffusion in one dimension. So single diffusion, only one person is moving. Actually, we are dealing with antigen antibody interactions, but here only the antigen is moving in one dimension. That means it occurs in a straight line. So the first picture you can see is the ring test. We have already told about that. Both antigen, initially you have antibody in solution. Above that you are layering antigen, which is also in solution. So in the junction of two liquids, at the junction of two liquids you will get a ring as formed. That's why the name is ring test. Okay, below that you can see another picture. So where it's describing about this single diffusion in one dimension. So initially you will take, so here you should remember that in this immunodiffusion it is carried out in agar medium. So initially the antibody is incorporated into this agar gel. Above that antigen is layered over, which is present in solution. So what happens? There will be interaction. Since it is single diffusion, so here the antigen only moves. Antigen moves down, and there where you have the zone of equivalence, there the precipitation will be occurred. Okay, to understand more, we have another picture. So here you can see that below the green color, that is where you have the antibody incorporated with the agar gel. Above that in the solution you have antigen. Now what happens? The antigen comes down and there is a ring of precipitate. Now there is another thing, double diffusion in one dimension. So this is also happening in one dimension, but you have the movement of both antigen and antibody. So here you can see in the test tube you are taking, initially you are taking the antibody is incorporated in agar gel, similar to the first case itself. But the difference is that now you have to add an intervening, you have a column of agar gel. Okay, you have a white column of agar gel. Above that you are adding the antigen is layered over that. Now what happens? Both antigen and antibody moves, and in this intervening column of agar gel you get a precipitation band. Okay. Now we will discuss about the third one, that is single diffusion in two dimension. So here so up to this we have dealt with a test tube and agar. Now in a flat surface you will take this antibody incorporated in agar gel. Okay, in a flat surface, and in that you will punch a hole or a well. In that well you will add antigen. So what happens? Since around the well you have the antibody incorporated in agar gel, so definitely antigen will diffuse, and the diffusion in how it occurs, it's not in a straight line, it is radial. Radially, just like a halo it appears. Okay, so that means it is in two dimension, it occurs in a plane. So the diameter of this halo, that is proportion to this concentration of antigen. If you increase the concentration of antigen, there will be more big halo will be formed. We have a beautiful picture. Here you can understand that the halo is formed. So in the well you have the antigen which diffuses, and it is forming a halo such that where you have the zone of equivalence, that is optimum antigen antibody concentration, there you get this, the halo. You can there is a marked, means we can identify that halo there. Okay, now we will discuss about double diffusion in two dimensions. So double diffusion in two dimensions, when you are saying that here it is double diffusion, that means both antigen and antibody will move. So initially what you have done in single diffusion in 2 dimension, the radial, it's also called as radial immunodiffusion because it happens in a radially it's going, just in a plane it is happening. Okay, so in that we have incorporated antibody in agar gel, but here in double diffusion in 2 dimensions, what we are doing, we are taking plain agar where you have punch out so many wells. In one well you will add antigen, in one well initially you will add antibody, and surrounding this antibodies well you have several other punched out holes where you add antigens. So what actually happens, if the adjacent to this antibody, if you have identical antigens, then the line of precipitation, they will fuse each other. Definitely, since the antibody is there, surrounding that you have antigens well, so definitely there will be precipitation band. But here what does actually said, these precipitation bands will fuse each other if they are identical, if the antigen are identical. Now if the antigen they are not identical, what happens, instead of fusing they will just cross each other. Now what if they are partially identical, there will be a spur formation. Okay, so that is happening in double diffusion in 2 dimension. So this is the picture which we have already shown for. First one is single diffusion in 1 dimension, double diffusion in 1 dimension, single diffusion in 2 dimension. This is double diffusion in 2 dimension. So the first picture you can see, that is fuse each other. That means antigen, adjacent antigens are identical. Next one is cross each other. That means adjacent antigens are not identical. Now you have a spur formation. That means antigens are partially identical. So this is usually we use whenever to detect different antigens. Just like in case of detecting some different antigens, we will use this technique. And radial immunodiffusion is usually used in case of influenza.